Hello, everyone. My name is Carl. I'm actually I'm just a guest here. I'm a PhD candidate in the MIMO group at Norwegian University of Life Science in Oslo, Norway. I decided to move back to Denmark. I'm Danish because my girlfriend and I had our daughter half a year ago. So now I live in Copenhagen, even though I'm employed in Norway. So I was really happy that it was possible for me to become a guest here at CEH because I work with hologenomics or worked with hologenomics in my PhD. So I don't want to bore you with my complete PhD thesis, but I want to present one of the parts, one of the papers that represent part of my thesis, which is this Comparem2 pipeline. So Comparem2 is a software tool that so a name is never a good name if you have to explain it right, but you can compare them because you can compare the microbial genomes. So let me get into it. So this was an internal presentation at first, so that's why I have a lot of animations. I assume that almost everyone here will be working with microbial genomes from prokaryotes like bacteria or archaea on a daily basis. Is that true? Can I see a raise of hands? A few, half, a third maybe. Okay, so this is a pipeline that is really only for prokaryotic genomes, right? So yeah, so if you work with hologenomics, a big part of it will be, I don't have to explain hologenomics, but a big part will be the microbial side. So you will be having to analyze and compare these microbial genomes. So if you imagine having any microbial genome from a prokaryote, it may be from a cultured isolate, so that's a de novo assembly, or it might be from metagenomic bins from a complex environment. In any case, you want to be able to compare them. And what I imagined having early on in my PhD was to have a pipeline that made me able to analyze these MACs and isolates in an easy way. So if you've been facing the same challenge, having to analyze microbial genomes, you will have been thinking about using these different concepts or tools. So you will have been looking into doing quality control, annotation, taxonomic analysis, and so forth. And you might have been using some of these different tools that I'm outlining here. So you stumble upon a tool on the web that solves your issue. Maybe you want to look into horizontal gene transfer or GC content, GC skew, special annotation, phylogenetic analysis, anything where you can analyze a genome on a genomic level. And you find a tool online and you try installing it. Sometimes it works, sometimes it doesn't work. So some of the times you will get into these kind of like weird situations where you're trying to install a tool, but you end up having like different issues and it doesn't always work. And that's what we call the dependency hell, which is actually a real thing. And the documentation of many tools may be lacking, so it may be hard. It might be hard to debug your issues. Conda has really come a long way. Even since I started my PhD three years ago, Conda has evolved a lot. So a lot of things are getting easier to do, but we still have challenges with Conda. If you put the wrong channels, you get into trouble. You can also work with Docker images that kind of encapsulate all the software into like one static image. But then you have to think about databases because they usually don't come with the Docker image. And then half a year later or a year later, it's game over because your version is now outdated. You have to start all over. Also, if you've been using, I don't know if anybody in here has been using K-base or Galaxy to analyze. I see some hands. So you can't really trust whether those services are online if you don't have control over them yourself. So my solution to this, and the bottom line is, so 9 times out of 9, you just want to gain biological insights from your microbial genomes, right? You don't want to be messing with like technical issues. I find myself very often spending a lot of time making things work while I actually just want to do actual research, right? So if there was a way that all these different tools that we all use to compare microbial genomes, if they could just be part of one pipeline that kind of did everything for us, we wouldn't have to spend so much time working with the technical challenges of installing these tools. So my solution was to kind of start over and make one big pipeline, which is this Comparum workflow. It's based on SnakeMake, which I think is the best workflow manager. I'm using Aptena containerization to make sure that everything works together and you can download everything in one go. I made a simple and effective command line interface, so it's easy to use this tool. I published the tool on Conda, so it's easy to install, and it has completely automated database download and installation, and it's really easy to install because of the Conda packaging. And then on top, it takes all the results from all the different tools that it runs. It makes a visual report that takes all the results from the different tools and put them into one place where you can compare them in an easy way. So this is the complete rule graph of all the different tools that I put into this tool. So hopefully some of these words will make sense to you. So we see DGDBTK, Apricate, Eggnog, MLST, cake pathways, mass tree, Proca. Proca, that should be a big one. I think many people in this room have been using Proca at some point. Busco, Anti-Smash, Panaroo, Checkam2, also Bacta actually, and different phylogenetic tools. So these are all the tools that I integrated into this package. So how can you get it? Yeah, you can just install it from Mamba, or you can actually also clone it or fork it if you want to make a pull request to add stuff to it, because it's it's open source. And I wanna showcase this genomes to report concept, because I think it actually makes a lot more sense than I thought it would do in the beginning. So I want to show you how easy you can actually get started. So this is my terminal, and I asked Caroline, whom I'm sitting in the same room with, if I could borrow a few of her Macs to just kind of see what kind of data you're working with here at CEH. And this is just a small list, but it will do. These are 8 Macs that Caroline had. Can you make it a little bit bigger? Can I make it larger? Of course. Let's see.
Do you need glasses? Oh, we have glasses.
So this is this is just 8 Macs. These are recovered from the guts of wild salmon, and so they are from mesogenomic binning. And these are just the bins that come out that represent different species. So what we can do is we can just activate the environment, and then we can run it, the tool, Comparum 2. And then, because I don't want to have you sitting and waiting for a long time, I'll just run a fast analysis, completes in a few seconds. So it runs the tool, makes a list of the Macs, builds a graph, and starts running the tools. And because it's using SnakeMake underneath, it's making sure to use all the threads on your machine. So if you have 64 threads, it will make sure that you run as many tools as you can concurrently. So now finally we see this is the full list. This is the rule graph that I showed you earlier in the presentation. You see some of the tools have completed because they are part of the fast run preset. So now it actually creates this HTML file, which is a visual file that you can investigate. So here it is. You see it's produced today, the 4th of December, 11:44. That's right now. So these are the Macs again. And now you see in the report, it shows you what analyses have been computed. And these are just a few of them, because I don't want you to sit and wait for a long time. And it gives you a visual interpretation. So already now you can see this is a figure that shows you the context and the DC content. So it's already becoming palpable what your genomes kind of look like. And it also computes a mass tree, because why not? It's nice to look at a tree and see the phylogenetic relationship. But you can also run the complete pipeline if you have a bit more time. So I did that. This is the complete report. If you run all the tools in the complete rule graph. So what you see here is it starts out the same way. You get the same figure with the context sizes, but now you also get Busco. So that is a type of quality control where you look at the completeness of genomes. And you also get Checkam2, so you can kind of compare. If you get better completeness with Checkam2, maybe you want to use that, right? and you can also see your contamination. And then it goes on, it gets a bit more advanced. So you have multi-local sequence typing, which is more of a clinical thing, actually. I'll go further. And then we have Abricate, which is also clinical, but it's a really fast tool. So I thought, why not incorporate it? And it's kind of striking to see that. So what it shows is it shows you the antimicrobial resistance genes that are present in your genomes. So I asked Caroline, why are we seeing 6 different antimicrobial resistance genes in one of the MACs? Because these are wild salmon, right? That's kind of weird, but apparently there's a spillover. I think it's been known for a long time. But this just shows you that if you didn't look into this analysis, you wouldn't gain that insight. So it's actually quite useful to run a lot of different analyses, even though you don't necessarily want to look into antimicrobial resistance. It still gives you some insight. Also, virulence factors, lots of those in the same genome. You get your PROG annotation. Of course, you can't really show an annotation in a small table. So these are just the number of genes that are called. You also get EGNOG annotation. So you get your K-SIMEs, EC numbers, gene ontology numbers, KEGG pathways, PFAMs, and you even get a heat map showing you the number of genes from different COG categories. So you can kind of compare. If you have several MACs from the same species, you can kind of see, is there some of the MACs that has more genes from a specific category? You get pathway enrichment analysis, which is just a way of looking into whether the genes from a specific pathway in your genome are overly abundant or overly present in your genome. So here you also see different number of significant groups of pathways that are present in the genomes. And all this data is actually available here. So you can start running through the pathway enrichment analysis, and you can start already now making hypotheses. You can generate hypotheses about the differences between these MACs already just from looking at this report. And there's even an Excel button, so you can download an Excel sheet. So you can kind of quickly get on with analysis. It's also dbCAN. This is carbohydrate active enzymes. So here you see we only have 6 of the MACs actually being present with different carbohydrate active enzymes. So that tells us that 2 of them are missing because we had 8 MACs, right? And those turn out to be mycoplasma. So you can also see, we can say something about what species are doing what in the guts of these salmon. You get a pan-core genome. So what we see here is even though some of these MACs are actually the same genus or even species, they don't share any genes because the core genome is 0. So that tells us something about a lot of uncharacterized variation in these species. And of course, you also get the species. So number 2 in the bottom, which was the one with a lot of antimicrobial resistance genes, that's a Pseudomonas alloputida. And the 2 that are missing from the carbohydrate active enzyme list with the mycoplasma, as you can see here. And you also get the phylogeny. So what this report is doing is just integrating all the different analyses into a single place where you can quickly compare different aspects on different levels like quality control and annotation and phylogenetic comparisons. So let me get back to the presentation slide. So in the future of comparing 2, I will add more tools because I use this on a daily basis because I work a lot with microbial genomes. So I will add even more tools, and I really like DRAM, but it's really hard to use DRAM. So I want to incorporate some of their heat map visualizations into the report as well. I also want to add anti-smash, which is partly working now, but will be working even better in the future. And then I also wrote a documentation because I really want to get people into using this, because if this tool had existed when I started my PhD would only have been like half a year. I'm just kidding. But I think a lot of time goes for PhDs to just kind of do these basic analyses and like getting tools to work. So I really want to get people into using this, and I really hope that in the future there can be like a open source culture where people are like adding tools and making pull requests. But that's of course very naive. So yeah, I just want to say thanks a lot for hosting me here at CEH, and thanks for listening to our presentation. If you have any questions, I'll be very happy to answer those.